is Noelia Anton Bolanos from doing work on chimeroids from the lab of Paula Arlota. And so now not only using multiple lines, but using multiple lines in the same dish together. So a whole new ball of wax. We're interested to see what you have to tell us. Thank you. And I'll stop sharing so you can start, Noelia. Looks good.
Now, you can hear me, you can see the screen.
Perfect.
Yes. So first of all, thank you so much. I'm very happy to be here today and share some unpublished data that we have in the lab. I just want to mention that this is the hard work of me and Irene Faravelli, that we have been joining forces together to work together in this project. So basically, when we started the project, we really wanted to ambition new models to be able to assess these problems that we are speaking today about, like how human inter-individual variability can really affect how each of us can respond totally different to neurotoxic stressors. We can think about perturbations, like genetic perturbations, like Alex was speaking before, but we can also think about environmental perturbations of any other perturbations. So the overview of the talk, first of all, I'm going to speak a little bit about the human cortical organoids that we have been working for many years in the lab. I will jump into the chimeroid concept that I want to share with you today. And then at the end, how we are using these chimeroids to really start to understand the inter-individual variability in response to neurotoxins. So I don't think I need to really explain too much in this audience why we really need to start using these human models. So you can see this in this simple drawing, how we are very, very different from animal models. Our brain is encephalic versus the mouse that is encephalic. We also have different molecular logic in how we are generating the cell types, even though that there is a lot of convergence, but also we are making many different cell types. So it's very important that we start to work in these models, but not only for that, also we know like we have very limited access to human brain development, because mainly happens in the womb, in the mom. And also, if we want to really start to study complex genetic disease, we really need this genetic makeup that the human have. Like, it's very different from any other animals. So for that, human brain organoids can really help us. And in the lab of Paula Arlota, for many, many years now, we have been working in trying to produce human brain organoids as reliable, as reproducible as possible. Very specifically, we are very focused in the development of the cerebral cortex. And this started like 7 years ago with Georgia Quadrato and followed with Silvia Velasco. And basically, I don't think I need to explain in this audience how we, from pluripotent stem cells, we are going to give a specific patterning for dorsal telencephalon. And with these pluripotent stem cells, we will start generating neural stem cells and neural progenitors that are already like patterned towards dorsal telencephalon. And at the very end of the differentiation period, we are going to start to have all these different cell types that we can find also in the endogenous tissue. So the work of Silvia Velasco, some time ago, she really saw for the first time that using this protocol that she set up, so basically we can have different batches and different replicas that are exactly by each other. So we were achieving like highly reproducibility with these models. But later on in the lab, Anna Uschiano and Amanda Kedagli, they decided to put together a full developmental trajectory of the human development from 23 days to 6 months. And what they saw is basically this reproducibility was maintained along all these different time points, that we generated the expected cell diversity very similar to the endogenous brain. And of course, even though these brain organoids are growing in a culture, metabolism was not really affecting the molecular identity of these cells. So we're very happy about all these results, and we thought that it was actually the moment to jump into modeling ASD. So this is not my work, is the work of very talented postdocs of the lab. You can see here down. So basically, they took 3 different mutations that are associated with ASD, autism spectrum disorder, and they compared this mutation to isogenic controls, using many different techniques to envision to reply to some of these questions that you can see in the screen. But because of time, I don't want to enter into this paper. This paper is published, but I want to point you to a very specific data. That is what really Irene and I were starting to think about it and how we can solve this. So basically, you can see these GABAergic interneurons that go like we have a huge amount of these GABAergic interneurons when we mutate specifically SUB, but you can see this in these 3 different donors, but you can really appreciate that the penetrance of this phenotype is different depending on the donor that we are looking at. So this really brings like a question that actually probably genetic background is modulating this penetrant in these mutant phenotypes. And also, of course, that there is a need in the field to really assess this, how what is the effect of human genetic variation on these cellular phenotypes, and if we can do it, we can do it at a scale. So this basically is how this Chimeroid project started. So here you can see here Irene Faravelli. As I mentioned before, we are working together in this project, and Tyler Fates, that is the computational biologist that's helping us with all the analysis that I was going to show you today. So what we were envisioning with Chimeroids is like if we can put a population of people together in the same entity, that is how we call these Chimeroids. And after putting all these donors together, if we can now apply different stressors. And by stressors, we can think about, as I said before, genetic perturbations, drugs, infections, of any environmental perturbation that you can think about it. And our main response is going to be a molecular response. It's going to be based in single cell data. The very good thing about this Chimeroids is it's very flexible, not only in the stressors that you can apply, also the different cohorts of patients versus control, of different polygenic risk score that you can really apply to the model. So we started the project doing what many other people are doing in the field. So we were thinking that it was going to be simple to take pluripotent stem cells from different donors and putting together at the very beginning of the patterning period. So basically, then we go through the patterning period, and at the very end, you can see here at 1 month, we have the canonical organoid that we usually generate in the lab, but we were envisioning that we will have all the different donors inside. But we did some low passage whole genome sequencing, and you can see here the abundance of different donors. You can see here the different mixes. And you can really appreciate in mix 3 and mix 4 that actually one donor was basically taking over the culture. So Irene and I, we were thinking, okay, maybe we can take out this donor. For some reason, it's very proliferative, it's taking over the culture, but you can observe in mix 1 that then other donor was taking over. And if we take out the mito210, then we lose another donor. So basically, we conclude after doing this test that there was no way to maintain the balance of these different donors when we are starting from pluripotent stem cells. And this is something that the field has been experiencing when you really want to put together different cell lines in the context of brain organoids. So then Irene Faravelli and I, we came up with the idea of, instead of using pluripotent stem cells, why we are not going to use a cell type that is more stable, like neural stem cells, and how we did that. So basically what we are doing is we are going to generate brain organoids, like cortical organoids of each of the donors, and we are going to do the patterning period individually. And at the end of the patterning period, we're going to dissociate these organoids and reaggregate them as neural stem cell chimeroids and let them mature. So I don't think we should take this as a given. We were very surprised that actually the structure of the typical structure of brain organoids was maintained. You can observe here at 1 month, but also at 3 months, we were observing like the typical canonical markers that we can observe in 3 months organoids. Then we ran some low passage whole genome sequencing to see the balance distribution of the different donors. And you can observe that now we will actually be able to maintain the different donors. Then we ran single cell at these 3 months organoids, and you can observe here, we were very happy to see that actually all the different cell types that we were expecting to have at 3 months, they were there. So we were not skewing the populations by doing this dissociation reaggregation process. And also we plot on top of this UMAP, the SNP distribution of the different donors. You can really observe here that they are fairly very well distributed along the UMAP. If we then multiplex this 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 UMAP here, you can observe in this mix 1 and mix 2 that all the different donors were actually contributing very similar to all the different cell types. And this is like, we were extremely happy because now this means you can really use this system. Then if we want to think about these lines as they are like very proliferative, or we have sometimes some problems to differentiate them to dorsal telencephalon. And of course, in the field, we don't want to like stop using some lines that are maybe going to skew this population. So we really want to be agnostic to the stem cell that we are going to use. We did a test with this GM that is here in this blue color. And then you can observe that this gene was highly proliferative. It was almost taking over when we were doing the neural stem cell chimeroids. If you observe here in the pseudotime, this GM line has a huge amount of cells at the very early stages of the pseudotime, meaning that we have a lot of proliferative cells, and that's why probably this line was taking over. So we were thinking of how we can overcome this problem. And we decided to push a little bit more further into the development of these progenitors. And instead of using neural stem cells, now we are going to use neural progenitors. So what does it mean? It means that instead of mixing at the end of the patterning period, we are going to keep during the differentiation period. And at the middle of this differentiation period, we are going to dissociate them and reaggregate them again. And now it was really a surprise that actually we were able to make organoids with what we call a neural progenitor chimeroids at 3 months, and we have the populations that we were actually expecting at these 3 months. And you can observe here, if now we compare this stacked bar plot of the different donors versus this one, you can really observe that now the GM was more controlled inside of this chimeroid, and we are actually making the same cell types on all the different donors. So the next question that we wanted to answer to be able to use this model is if actually, because we are going through this dissociation reassociation process, if we are following the proper developmental trajectories. So basically what we wanted to test if our organoid, the typical organoids that we are making in the lab are actually very similar to these neural stem cell chimeras or neural progenitor chimeras. So we took our single cell data and then some different transcription factors that we know that are very specific for all these different cell types. And we observed that actually, if we compare the atlas versus the neural stem cell chimeroids versus what we call single donor neural stem chimeroids, that is like, instead of mixing all these different donors together, we are going to mix them only with itself. So it's kind of a control. And then we also have here at the end the neural progenitor chimeroids. And you can really observe that basically the levels of these transcription factors in these different cell types, they are mostly the same. We also did some transfer label to really see if the different cell types that we have in our chimeroid versus the atlas organoids, if they actually correlate with each other. And you can really see here beautifully that actually we are generating exactly the same cell types that we are generating our single donor atlas organoids. We also check a reproducibility measure that is adjusted multiple information. And here in the dotted lines, this is the variability that we are expecting in fetal tissue of different data sets that are available. And you can observe that actually the atlas and all the different conditions that we are working, our single donor chimera, multi-donor chimera of different mixes and batches, that actually the variability is not higher, the variability is not higher as the variability that we expect in the fetal tissue. So these are great news because this means these chimeras are as reproducible as our canonical organoids that we were generating in the lab. So basically, as a summary of this first part, I just told you that we are able to combine lines and maintain the different population of donors if we mix the cells, either a neural stem cell stage of a neural progenitor cell states, and also that these chimeras are very, very similar, if not identical, to the organoids that we have been generating in the lab. But now, after having all this, these are great news because we are able to really use these chimeroids for actually the purpose of the project, that we really wanted to dissect their individual variability and susceptibility to neurotoxic triggers. And we chose two different neurotoxic triggers. Very specifically, we chose ethanol to mimic fetal alcoholic spectrum disorder and valproic acid because it's known that actually is taken during pregnancy, the offspring can have ASD. And we know that in the clinics, these two disorders have high phenotypic variability. So we really wanted to be able to assess if we are able to catch up this variability using our chimeroids. So we treat our chimeroids with ethanol and with BPA. And you can see here with a single cell, this is the control, the ethanol and the BPA. And you can, this little cluster was a new cluster what was appearing in the BPA case. And you can observe that these are mature interneurons. And if you remember from the beginning of the talk, when in the lab we're actually mutating three different, making organoids from the three different mutations that generate ASD, we actually were having an increment of this also interneurons. So it was very nice to see that either if we use genetic perturbation or a neurotoxic perturbation, we were actually getting the same phenotype. So it was a very nice result. So basically, this is just to show you that how the different mixes and replicates look when we are using these chimeroids. This data is barely batch corrected. They are basically identical to each other, which is beautiful. And then basically, if we take a look at how the different cell types are, the balance of the different cell types are changing when we are either in the ethanol in purple on in the BPA in this multi-donor chimeroid, you can observe that, for example, we have these interneurons upregulated. We also have some decrease of CPNs, increase in the case of the ethanol. If we take a look now, instead of using multi-donor chimeroids, we were using single donor chimeroids, and then we multiplexed together bioinformatically to see if we were able to see the same results when we are using a multi-donor chimeroid versus when they are actually growing individually in the same individually in a chimeroid. So in general terms, we were observing most of the effects that we were observing. For example, you can see here these interneurons in the BPA case. Also, the CPNs were going down. So most of the changes were maintained, and we were very happy to see that. Then if we want to take a look at what are actually the specific effects in the different donors in the BPA treatment, here are the different data sets. You have here the control, the ethanol, and the BPA with the different donors. And one observation that you can see here is that actually this line CW was having a donor-specific effect, but also treatment-specific effect. You can see that the BPA, this line was being more abundant in the case of the BPA and less abundant in the case of the ethanol. So it was the first hint that actually, even though that these lines are growing together in the same entity, we were able to dissociate this specific effect. If we move forward and look at specific cell types that were actually statistically different between BPA and controls, you can observe specifically, for example, in the outer radaglia, how the CW was going up. The outer radaglia is going up versus in the 11A is going down. You have, for example, a different effect in the case of the CPN, in which the CPNs go down in the CW, and they go even more down in the 11A. In this case, we observe a case of difference in penetrance. It was very nice to observe these differences between the different donors. One of the last things that we did was actually we took our single cell data and we pseudo-bulk our single cell data and plotted in the PCS space. And what we did is to measure the Euclidean distance between the controls and the BPA. So you can see the BPA in triangles and the controls in circles. And then by measuring this Euclidean distance, we were able for the first time to stratify the different donors between less affected and most affected, as you can see this in this graph. This is the global effect of all putting together all the different cell types, but we can also separate them in what will be the effect in a specific cell types. And you can observe that independent of the cell type that I'm showing you here, you can, for example, see that in the case of the outer radaglias, we have 11A that is going out versus the others that remain in the same bucket. So this is like the end of the talk. And as a summary, at the beginning, I show you that we are able to maintain a balance between the different donors. If we go ahead and use neural stem cells or neural progenitors, these chimeras are very similar and follow the same developmental trajectories as the normal organoids, and very similar also to fetal tissue, that we are able to actually model individual susceptibility when we are using these multi-donor chimeroids. And at the very end, I show you for the first time that we are able to stratify the different donors that are affected more or less differently when they are treated with BPA. So with all this, I really would like to thank you for your attention, and very especially Irene Farabelli, because we are working very hard together in this project together, and also everyone in the lab that has been helping in the project, very especially Paola Arlota, our mentor, all our collaborators, and all the funding agencies. Thank you so much.
Thanks, Noelia. Any questions?
Hi, Novin from Kristen's lab at Yale. Great talk. Thank you very much. Just quick question. Do you think if you increase the concentration of or the concentration of VPA or ethanol, you can push all those donors to the same direction? It's not concentration dependent. It's just depend on the genetic background of those donors.
This is a very good question, a very interesting point. I bet, I mean, with the BPA, we might maybe push them, but I don't have the answer. With the ethanol, I think we might find more differences between the donors. Probably we increase the concentration. But it's a very, very interesting point. Maybe something what we should try out in the lab.
Thank you. Thank you very much.
I have a quick question. In terms of using this model system for studying disease, is that something you would recommend, or is there a concern that there would be crosstalk between disease cells versus non-disease cells?
Of course, non-cell autonomous, this is a very great question. First of all, thank you so much for pointing it out. Of course, non-cell autonomous effect can influence. That's why we're doing this single donor, to be assured that we can use the model for that. Our hypothesis is that if your phenotype is very nuanced, it can be masked. But if you have a very strong phenotype, probably you are going to be able to read it.
Thank you.
Thank you very much for this talk. It's really interesting, and it's really like one of the approach that we are allowed to really study a lot of individuals, and this approach to limit the influence of like cell division is really important. What do you think, like using these different steps in which you mix, like what do you think is the amount of individual that one could reasonably mix together? Have you tested like mixing more and more different IPS line together?
Yes, basically this project started like as a pilot experiment in which we wanted to maintain like simple, to be able to actually do all the individual donors and, to really assess very well what we are doing. But now in the lab, what we are doing is really like increasing up the amount of donors that we can put together. I guess the limiting factor is the beginning. So how many how many cells you can sit together? We have been making a lot of testing to increase the amount of cells that you see at the very beginning, to be able to even increase more the amount of donors that you can put together. But of course, we are very interested in scaling up.
Go ahead, Alon.
Hi, thank you. That was very interesting. So I'm curious, when you were talking initially, when you're trying to use the stem cell level, every time one cell took over, I was wondering if you have a thought about what's the mechanism that made it happen then and didn't make it happen when you were using the more differentiated versions.
So this is a very nice and interesting question, and I think it's a question that the entire field is probably asking themselves, because we know pluripotent stem cells are very variable and biased towards specific fates. We don't know it's coming from the reprogramming, from the epigenetic memory of the cells. We still don't really know if maybe instead of using more naive cells, it will help this, because the different pluripotent stem cells are going to react differently to the small molecules that we are adding. So this also generates biases inside of the plate. So what we were trying to do if using a cell type that is actually more controlled by the developmental speed of the species. So basically neural progenitors, if they are well patterned towards dorsal telencephalon, they are going to have a specific ratio of proliferation that is basically controlled by, but that's why we are not going to have, like, I mean, Alex was speaking about macrocephalic and microcephalic. So basically it's more or less controlled by the species. So we wanted to use a cell type that is more controlled. Basically, this was the main goal.
Thank you.
Thank you so much.